Hello and welcome back to another Starting Out with Quantum Computing video. I'm your host Lloyd and I hope that I've got an exciting one for you. So it has been a while and I have been very busy, but I'm very glad, very glad to be making another podcast, article, video, whatever format you like to consume these things. We're back. So we're talking today about a development in the field of quantum computing, in the field of quantum chemistry, in the field of biology as well. There's quite a few intersections here. And this happened primarily last month, but I was so interested in it that I wanted to at least speak about it. It is indeed the research breakthrough by Cleveland Clinic, Riken and IBM, as they simulate a huge protein using quantum computing. So this was announced on the IBM blog just a couple of weeks ago, and it was submitted on arXiv days before that. A large team of researchers performed arguably the largest atomic simulation to date. And if you don't know what arXiv is, this is a paper archive, namely, by Cornell University. This team represents an international collaboration. We had researchers from Cleveland Clinic in the US, IBM, both in the US and Japan, as well as the Riken Center for Computational Science and IBM Tokyo. Specifically, this team were able to cross the 12,000 atom barrier by simulating the protein trypsin, which is composed of 12,635 atoms. That is quite a lot. In this work, they also simulated T4 lysozyme, which is composed of 11,608 atoms. Being able to simulate large biologically relevant molecules, such as proteins, is incredibly important as it informs us of the underlying mechanisms at play inside our bodies. This in turn can further fields such as medicine or drug development. So let's dive into exactly what happened. So I mentioned two proteins. And this team of researchers, they used something in their research known as a quantum-centric supercomputing framework, or QCSC for short, to look at two specific large protein ligand complexes. So let me explain what these terms are. QCSC is a type of computing where you basically integrate quantum computers with your powerful normal supercomputers. And so you let the quantum computer handle the things that it is better at, and you give the supercomputer the tasks that it is better at. And this can sometimes give you incredible accuracy for certain tasks. And then you have the protein ligand complexes. Now, I'm not a medic, I'm not a doctor, but I can at least tell you that they are biological molecules formed when a protein binds with a much smaller molecule known as a ligand. And they are essential to life, these complexes, these protein ligand complexes, as they contribute to enzyme processes, our immune system, and the receptors in our bodies. Without them, our cells simply wouldn't be able to keep going. So the smaller of these two protein molecules that they simulated is called T4 lysozyme. It's made of 11,608 atoms, as I said before. And although this isn't made in the human body or isn't produced in the human body, it is a protein that is used commonly in labs for studying cells and bacteria. It helps to break down bacterial cell walls and can be used to extract proteins and DNA. It's a very useful tool for any biologist. The second and larger of the two proteins is called trypsin, 12,635 atoms. And this protein is made in the human body and is essential for nutrient absorption and digestion. It is a digestive enzyme that is produced in the pancreas, and it binds to proteins to break them down into amino acids. We most certainly need it to function. The team chose these particularly large proteins probably to make a statement. They wanted to show that quantum computing could be used to study molecules that are large and relevant to current science. They also simulated the binding of these proteins as they were immersed in a liquid water solution, which makes these results much more realistic to the internal conditions of, say, the human body. So let's take a look at some of the details. So months before, in March this year, IBM and Cleveland Clinic had partnered to simulate a mini protein consisting of just 303 atoms using this same QCSC method. Now, with the addition of Riken's powerful supercomputers this time, the team have been able to probe molecules that are over 40 times larger than the work in March. They also claim their results boast a much larger accuracy than before, at least in some parts of the overall workflow. The two 156-qubit IBM Quantum Heron R2 processors, that's the name of IBM's quantum computers, employed quantum sampling. It's a process where the quantum computer generates the complex data over time. As you let the quantum computer sample more and more, you start to build an accurate picture of what is actually going on in your system. Following this, the Japanese supercomputers Fugaku and Miyabi-G were used to process the resulting data. Just as before, the researchers relied on something known as wave function-based embedding. It's a process which breaks down a large problem into manageable chunks. They then feed certain chunks to the classical computers and certain chunks to the quantum computer. The quantum chunks would generally have more complex interactions within them, for example involving entanglement between many atoms. They would then use a quantum algorithm known as SQD, which is otherwise known as sample-based quantum diagonalization, to solve the chunk. Finally, the classical computers would stitch all the chunks back together to give the researchers the final result. But make no mistake, this process was a huge undertaking. Considering both proteins, the team needed 94 qubits across the two processors, running 9,200 quantum circuits for over 100 hours, and they ended up with a data set on the scale of around 1.3 billion points. I mean, that is huge. These numbers, for anyone who works in the field or even knows a little bit about the field of quantum computing, you'll know that these numbers are insanely large. Simply keeping 94 qubits remotely coherent during a project of this scale sounds unreasonably difficult. There's also a plethora of technicalities that cannot be simply overlooked. For example, unlike before, this new simulation doesn't just calculate the electronic structure of the sole protein on its own. It also considers the protein binding to a ligand within a water solution. Equally, in the chunking process we talked about earlier, it's very unlikely you can just arbitrarily choose where to chunk. You need to understand exactly where is best to perform this sort of simplification, this chunking, such that you preserve the inherent physics or the chemistry of said molecule. There were also many obstacles that the team had to deal with. That method of breaking down a complex system into manageable chunks doesn't actually scale that well. Meaning, if you were to apply it to a molecule double the size, you'd actually need 25 times more computing power to make it work. To counteract this, the team realized that at a certain distance from the center of their trypsin molecules, effects like entanglement and other non-local correlations were effectively dead and absent. This meant they could truncate, in other words, simplify their chunking approach, and still generate accurate results. Another achievement involved the team improving the algorithm SQD, calling their new approach Trim SQD. Generally, SQD can be really useful in quantum chemistry calculations, as one is able to leverage the quantum computer to represent huge electron configuration spaces. This is something a classical computer would inherently struggle with. Instead, we can use the classical computer to interpret the results and find the solution. However, searching for the solution in these huge spaces can be like the old analogy of a needle and a haystack. Trim SQD works a little bit better by breaking down the search area into smaller sub-areas. This in turn helps us to identify and filter the useful information from the junk. Oh, and by junk, I mean things such as decoherence, quantum noise, gate errors, these sort of things that create results that aren't expected. And so, if you want to learn more about that, I do have a very good article, I say so myself at least, on decoherence, noise, and all things faulty, the science behind what makes quantum computers fail. But let's talk about this result. So what does this result by IBM, by Riken, by Cleveland Clinic, what does it really mean? While this fusion of classical and quantum computation is rather elegant, in order to accurately and truly determine the electronic structure of a protein, we can't try to solve things in just a classical way. At the smallest of scales, quantum mechanics dominates the interactions, meaning that the best direction to head towards involves a quantum mechanical treatment of an entire protein. Although powerful classical computers are efficient in modeling certain parts of a protein, this combined QCSC approach pushes boundaries and asks the question of what role do quantum computers have in building and developing new and more accurate research. Not only does this piece of research represent international collaboration, which is great, but it also demonstrates that there's merit to combining quantum and classical computing together. And the research itself has much potential. By pushing the limits of what biological molecules we can simulate, it means we can discover new complexes and develop new medicines, or study the mechanisms to complicated diseases. These are a few of many things that we can do. It means that we are getting much better at predicting how biology works at the smallest scales, the scales where quantum mechanics is king. This won't just have a positive impact on healthcare and medicine. Fields like material science, chemistry, and physics will all benefit greatly from a hybrid quantum computing workflow. So although I don't usually like to predict the future, it seems clear to me that quantum computers are going to change society for the better. And this is one small example of that. Thank you very much for listening, as always, to Starting Out with Quantum Computing. I very much hope you enjoyed, and I also very much hope to see you next time. And have a wonderful rest of your day. All right, bye-bye.